So here's our diploid cell, which is a starting cell with two sets of homologous chromosomes, and we're going to use it to show meiosis. So homologous chromosomes are similar in size, and you can see we have two long ones and two short ones. They code for the same category but give different traits. So for example, the homologous chromosomes would both code for eye color, but one gives blue eyes and the other gives green. So the chromosomes will duplicate in the S phase of interphase, and they copy into two exact copies of sister chromatids which are connected by a centromere. In prophase 1, which is the first stage of meiosis, the two centrioles make up a centrosome, and those centrosomes move to opposite ends of the cell. The chromosomes start moving toward each other and lie next to each other really closely. So the long will lie next to the long and the short will next to the short. And this is called synopsis, when they get really close together. And when they're really close together, they form a tetrad. When in a tetrad, these homologous chromosomes will cross over, swapping segments of DNA traits. This will help contribute to genetic variance. Spindle fibers fully form out of the centrosomes and attach to the connecticores of the homologous chromosomes. These spindle fibers will start a tug of war with the homologous chromosomes, and they will start to line up at the middle of the cell during metaphase 1. The way that the homologous chromosomes happen to line up next to each other determines independent orientation, as the homologous chromosomes in a pair can lie next to each other in two different ways, which also contributes to genetic variance. In anaphase 1, the centrosomes will wind up the spindle fibers into shorter segments. This pulls the homologous chromosomes apart, separating them from the middle of the cell. In telophase 1, the cell will begin to cleave, appearing in the shape of an 8. Then it enters cytokinesis, and the cell will separate into two. Similar to mitosis, in prophase 2, spindle fibers start to form out of the centrosomes that are on opposite sides of the cell. The spindle fibers connect to the connecticores of the chromosomes and begin the tug of war again. The chromosomes will line up in the middle of the cell again in metaphase 2. In anaphase 2, the centrosomes will start winding up the spindle fibers, but this time the spindle fibers will pull apart the sister chromatids from each other. These will become fully fledged chromosomes at opposite ends of the cell.
In telophase 2, the cleave between these cells will begin to form. Then, in cytokinesis, the two cells will begin to divide their cytoplasms into two cells each, forming 4 haploid daughter cells.